CCAT? The thing to note is that, so at the top here, it's going to give you some basic information. So like it says, reads 14,932. In other words, this DNA sequence was read 14,932 times, so it should be pretty accurate. It's going to tell you other things like that it's not likely to be circular, there are no suggested repeats. One of the two that we got back is broken into 4 separate parts because they think that there are repeats in the sequence. You can analyze it if you want, but I'm not going to obligate you to. And the one thing that I will put the name that's associated with each one of these. You can analyze your own, or you can analyze a different one. The nice thing about this one is it's only 45,000. It sounds like a lot, yeah it is, but actually this is a smaller phage genome, okay? Do you have to analyze the whole thing? No, we're short on time. But stick to the question that you're trying to answer, okay? And try your best to do at least some of it. That's the goal. So bear with me as in the next couple of days as we kind of get this up and going and as we're working on this. But my plan will be Wednesday for us to be working in here on this. Take a look at a sequence and decide on one that you want to investigate before coming in on Wednesday, okay? And it would be really great, too, if you have the time to at least familiarize yourself with whichever software or program you need to use for your question. So if you're just trying to identify open reading frames, look at the information on OrFinder. So under phage projects, I have a tutorial on how to use OrFinder, and then I've got links to different programs that can help with different aspects. So Grace and Tori, you guys are looking for codon bias. You might want to look for particular codons and maybe identify like maybe the largest ones and say, are there codon bias in these top 10? And if so, are these tRNAs encoded in the phage genome? That's kind of a very simple analysis that you can do to begin performing that experiment. And we've got programs to do both of those things. That's kind of the basic information. I know it's a lot to take in, 45,000 base pairs at once, but we will begin to look at this together on Wednesday. What this means in terms of the take-home is that the part with your partner, I think I'm going to push it back. I'd like to actually have a couple of days to work in here before I make it due. So just that last part, the 15-point progress report on the project, I'm thinking of pushing back the due date to probably sometime early next week, just so you are aware, okay? Also, be flexible on when the poster is due. I'm working my best to try and push that back as far as possible so that we can still get them printed in time. Okay? Right now, as far as I'm aware, our scheduled time for presenting is during class time on Friday the 27th from 9 to 9:30. Why it's a half an hour, I don't really know, but that's what it is. And in terms of the grading, I've put up a rubric and also some just general information on poster, putting together a poster. That is up on Blackboard under the Phage Project tab. I'm also going to try, today is going to be the first day that I beg, to get some of the other bio faculty members to look at these as well, so that we can get a holistic view of grading, not only from me, but from them as well. So like Pete would be a good person to look at these posters. Okay? Should I ask Francie?
Yeah.
I actually did not think anybody was going to say that, Grace. I appreciate it. All right. Well, let's get back to Ebola then. The one other thing you may have noticed is I've uploaded slides for HIV. We're going to try to cover that one in 4 days, and there's not as many slides as there have been in the past. I think that one's going to be kind of a very broad overview because there's so much detail on HIV that we understand. And we definitely don't have the time to cover all of it, unfortunately. So where did we leave off? I think...
What do we know about replication mechanisms?
Thank you. right here, right?
Yeah.
So what do we know about replication and transcription of negative single-stranded RNA viruses? What are some key characteristics that have to take place? Has to go to positive strand, absolutely. So regardless of whether it's transcription or replication, the virus has to be able to produce plus single-stranded RNA. The way that, and that plus serves as a template for replication, or depending on what it looks like, it might serve as the mRNA for translation. Ebola virus produces monocistronic mRNAs. In other words, the mRNAs that it's going to produce are going to be, sometimes the term is subgenomic. It's not the full genome, it's only part of the genome. Other viruses, on the other hand, can achieve this in different ways. So influenza, for example, is a segmented genome. It's got 9 different fragments. Each of those fragments not only serves as a genomic chromosome in some ways, but it also serves as the template for mRNA. Ebola does this differently. It's got a 19,000 nucleotide RNA sequence. And what we're going to find is that this sequence, this negative single-stranded RNA, will be polymerized to produce plus single-stranded RNA. That's going to be the antigenome, or template for more replication. We're also going to find individual mRNAs being produced, each with their own cap and poly A tail. So even though this virus is producing its mRNAs in the cytoplasm, the mRNAs are actually going to look a lot like host mRNAs. And then you could view this as being advantageous. So that's kind of a precursor to what we're going to look at today. Yeah, so the mRNAs that it produces are going to contain a 5' cap. They're going to contain what's analogous to a poly A tail. So their mRNAs look a lot like the host mRNAs.
And they're doing this in the cytoplasm?
They're doing this all in the cytoplasm. And we'll talk about some troubles of that later on. Okay. 7 proteins in total. All right, so I put more detail up here than I probably care for you to necessarily know, but where the process of replication and transcription occurs is in what's known as inclusion bodies. Inclusion bodies are not well defined. And what I mean by that is an inclusion body is defined by the proteins that are located there. It's not an organelle. But the other term that our book has used is viral factories. These are going to be locations where replication, transcription, possibly assembly are all occurring. The major proteins that seem to be involved in this process are the L protein, which is the polymerase, VP35, which will come back to its role later, but one of its roles, or it has multiple roles. One of them is being involved in polymerization as well. It's described as the polymerase cofactor. And then NP, the nucleocapsid. One of VP35's roles is actually to serve as a conduit or a scaffold between NP and L. NP and L don't bind to each other, but they both bind to VP35. And you can determine this by immunofluorescence. You can determine it by immunoprecipitation. Well, one of the key characteristics is that VP35 helps to bring L to where NP is located. And NP, the nucleocapsid protein, is always associated with what in this virus? It's always associated with the genetic material. It's always associated with negative or plus single-stranded RNA. And so VP35 brings L to the location where replication is going to occur. And these inclusion bodies are defined by staining of nucleoprotein, or NP particularly. So if we look at what these look like, here's some immunofluorescence. It's not in color, but we've got just kind of shades of gray for this one. So the top here are over top and the bottom images are showing from 10 to 24 hours post-infection of 293 cells with Ebola. Where is NP localizing? And you can see that you start to observe some of these punctated dots early on at about 10 hours, but what happens from 10 to 24 hours? What observations do we make?
Proteins like around the outside.
Yeah, so there's proteins around the outside here, and that location is around the nucleus, right? The nucleus is somewhere in the middle here, if we were to stay in that. What are other characteristics? What are the qualities of those dots? Don't think too hard. I'm betting that Reed could answer this question. What's different between this one here and this one at 16 hours?
They get brighter.
They're brighter, the intensity is greater. What else? Now think quantity. There's more of them. Yeah. So the amount of these inclusion bodies increases, so does the intensity of them, indicating that a viral replication is occurring. And you can quantify percentage of cells showing inclusion bodies, and this is actually quite telling. This right here gives us an idea of when does biosynthesis for Ebola begin. And it seems to occur somewhere between 8 to 10 hours that we're starting to detect the process of replication by the formation of these inclusion bodies. So the very broad overview is nucleoprotein, which is shown here in green, binds, it is bound to the negative single-stranded RNA. I tried to include a yellow color here instead of green or red behind it, so that's why it looks like this. I apologize. So the negative single-stranded RNA is replicated into full-length plus single-stranded RNA, and that plus single-stranded RNA then will be replicated into many, many, many copies of negative single-stranded RNA, all performed by the same polymerase. And again, it's an RNA-dependent RNA polymerase. I'm going to skip this slide because I really don't find it important, this one here. So if you have this slide up, just cross it out. We're not going to worry about it. But we can look at evidence to continue to support that VP35 is required for recruitment of L to these inclusion bodies. So here's another immunofluorescence. On the far left over here, we've got NP being detected with some sort of antibody that's tagged with a red marker. And then in the green, we have L, the polymerase. And note the difference between the top and the bottom is minus VP35 or plus VP35. So take a look at those results for a minute, and then tell me about the localization of NP and L in the presence and absence of VP35. Alright, so top one here, without VP35, do they localize together? Not really. It seems like L is kind of all over the cytoplasm. Here's the nucleus. Whereas NP is still able to form these inclusion bodies. With VP35 present, what happens to L? co-localizes. Localizes to exactly where NP is located. So this is evidence then to say that VP35 is an essential member of the inclusion bodies. It's an essential member of replication. Without VP35, you would expect that replication wouldn't occur because you wouldn't have NP and L localized to the same position to allow for that replication and transcription to occur. That's what that evidence there suggests. Alright, that's it for replication. Replication actually, mechanistically, to think about it, is rather simple. What's more challenging is with Ebola virus understanding the transcription process. Okay? There is one thing to kind of point out here. I just want to include some terms real quick. So they describe what's known as a leader sequence in the Ebola virus genome. Kind of think of this like a promoter. Okay? There's a transcriptional start signal, which is the site where to begin transcription, and then there's a transcriptional stop signal. And this is true for all RNAs, that there's a start and some sort of stop signal. In eukaryotes, the stop signal is a run of T's, which would encode for a poly-A tail. Okay? Transcriptional start signal has a very unique sequence, or a fairly well-conserved sequence, but we're not going to worry exactly what that is. So this is monocistronic. There are 7 genes, but there's only one leader sequence. There's only one single promoter in Ebola virus. So the question becomes, how does Ebola produce NP, VP35, VP40, the glycoprotein, VP30, VP24, and L? How does it produce individual monocistronic mRNAs from one single promoter? And the answer, or at least the short answer, is each of these has their own transcriptional start signal and their own transcriptional stop signal. And what happens is that the polymerase just continues to read and start and stop transcription at the beginning and the end of every one of these genes. That's the short story. So I'll repeat that. One promoter located over here at this 3' leader sequence. We have a transcriptional start signal, and the red bar here represents a transcriptional stop signal for every single one of these genes. Note that in some cases they're overlapping. VP35 and VP40 have an overlapping start and stop signal. I'm not going to have you worry about that, but just note that some of those do exist in here. Okay. Transcription stops at a run of U's. That run of U's produces what? What's complementary to U in RNA codes? That's our poly-A tail. So it produces this poly-A tail, this short poly-A tail, by basically stuttering on a run of U's. And this is not uncommon. A run of a single nucleotide over and over again is going to lead to slippage, stuttering, insertions, deletions created by an RNA polymerase. So this is just going to kind of create a poly-A tail. And the the polymerase L just kind of continues to transcribe all the way through this, but doesn't always make it to the end. Okay? So if transcription is always going to start right here, it's always going to end here, it doesn't always progress to the end. What does that mean about the relative abundance of NP versus L? Yeah, more NP. Absolutely. And the least amount of L. And that's exactly what we find. And this should make some sense. NP is the nucleoprotein. It's the capsid protein. We need more of that than anything else. I liken this to Ninja Warrior. Okay? So in Ninja Warrior, everybody starts out at the same beginning start site. Most people make it through the jumping from one platform to the next, and then some people don't make it through the rolly thing, and some people don't make it off the wall, right? So you start out with like thousands of participants, and maybe 30 of them make it to the end. That's kind of how I view this. Transcription's always going to start from the same point. We're always going to get NP made every single time transcription occurs, but at some reduced amount we'll get L produced. Jonah?
So what's the transcriptional start sequence then? Like, how is that different from the promoter? It's not gonna...
So the promoter is just where the where the RNA polymerase would bind, right? Let me put this in bacterial terms for a minute. The -35 and the -10 sequence where like sigma factors bind, that's a promoter. And then transcription starts at about 10 nucleotides downstream of the -10 region. That's the transcriptional start site. So the promoter is where the ribosome would attach to. The transcriptional start site is just the sequence where it starts to lay down A, G, T, U's, and C's, complementary to the template. Shania.
So when say like it's at the NP and it's at the red at the end.
Yeah.
And so it doesn't get kicked off. So how does it like cleave in between all of them?
It doesn't actually cleave. So what happens is it recognizes that transcriptional stop sequence. It'll lay down a string of A's, and then it just kind of stops. And to be honest, how it transitions from one to the next, I don't totally know. I don't know if anybody really understands that answer. But it's not necessarily cleaving these RNAs. It's starting here, stopping right here, continues to scan, and then starts transcription again at the next transcriptional start signal. So it's not as if it's producing a 19,000 nucleotide mRNA. It's producing one of about 1,000 and another one of maybe 800 and so on and so forth. Does that help? Okay.
Do you know what makes it randomly fall off? That causes there to be like less...
Why it just kind of falls off? I think that this is true pretty much for all polymerases, as my understanding goes, that that we say they're progressive, that they will continue on for a certain amount of time, and then just somehow the attachment of the polymerase to the template and the other proteins that are associated with, just they dissociate from each other, which allows for the polymerase to fall off. The rate at which that occurs, what's actually required, I think VP35 might be required for that, keeping it on there. I don't know exactly all the mechanisms, and actually this has been one of the really hard areas of Ebola virus. We just don't know because we don't have great models for this. This paper that this one came from is maybe 3 or 4 years ago, and at that time they didn't have a clue. So you could argue there's an evolutionary selection here. Most abundant protein, you want to put that at the 3' most end of your negative single-stranded RNA. One that you need the least of, put it at this end over here. So yet another mechanism by which gene expression gets controlled. One other thing that seems to be required for transcription is the VP30 protein. VP30, so this is that part of, actually within the leader sequence, so we've got this hairpin loop structure here, more nucleotides, and then this right here is the transcriptional start signal for NP. So that hairpin loop structure that we just looked at would be located right here where that arrow is. And what's true about that? Nope. Apparently I'm now going the wrong way. Okay. What's true about that transcriptional start signal? What's it located in? It's located in the hairpin loop structure. Okay. This right here, since it's folded upon itself like this, the RNA polymerase can't read this. It's actually occluded from being read by the RNA polymerase. And this is where VP30 comes in. VP30, as far as we know, will bind to, we describe it as an anti-termination factor. But basically what it does is it binds to this RNA here and helps to melt this hairpin. Take it from a hairpin loop structure and now allow it to unfold to single-stranded RNA. That now exposes the transcriptional start signal to allow for transcription to begin at the NP protein. If VP30 is absent, this hairpin folds back up, and we get no transcription of NP. We also get no transcription of any of the other genes as well. So one of the things that seems to be involved in the transition from transcription to replication or vice versa involves the presence of VP30 to control that process. I bring this up not to know the details so much, but just to highlight that there's a lot of complexity in the ways that gene expression is controlled. We've talked about with HPV, it's dependent on splicing factors. We've talked about with bacteriophage that it's dependent on sigma factors. And here's yet another example. Virus has all of the proteins that it needs to perform this process, but it's based on the abundance of those different proteins that's going to dictate, and their location in the genome, that are going to dictate the gene expression. Jonah, do you have a question?
Is VP30 included in the viral protein?
You mean like in the viral particle as it's coming in? Yes. Yes. So you might have some inclusion bodies that are performing replication. You might have some that are performing transcription. You might have some that are doing both at the same time. Let's come back to a topic we've talked about before. This is an RNA virus. At some point during this process of transcription and replication, what's going to be formed? Well, the endolysosome would come beforehand. But think about the process. If we have RNA and we're laying RNA on top of it, we've got what? Double-stranded RNA. Triggers some sort of antiviral response in the cell. In Ebola virus, what this seems to trigger is an interferon response, or IFN. And what we're going to spend one of our last 2 lectures talking about with Ebola virus is how there are different proteins to actually block that interferon response from occurring. So something about the virus inherently will trigger it. The virus has a way to inhibit it. The other thing is that since the RNA is constantly bound by nucleoprotein, or that NP, it actually will minimize the ability of the host immune response to recognize this double-stranded RNA. Usually the host is recognizing free-floating double-stranded RNA, but we rarely have that occurring, or it doesn't occur that frequently in the case of an Ebola virus infection. The major proteins that are going to be involved in this are VP35 and 24. We're only going to talk about 35 when we come back to this, because there's some really cool things that 35 does. All right. Last steps. 24 or 48 hours into infection, we get budding. So this is one of the first viruses that we've seen that don't exit by some sort of lysis mechanism. This is the virus here, we've talked about, enters through receptor-mediated endocytosis of some sort. The exit strategy is through budding. And the protein that's essential for this is VP40, which functions kind of like a scaffold protein. In the same way that VP35 scaffolds NP and L together to form those inclusion bodies, VP40 serves as a scaffold, and it actually will localize itself on the interface of the cell membrane. It's going to bind to NP. NP brings along with it VP35. VP35 brings along with it L. And we get other proteins as well beginning to kind of glom on to the interface of the cell membrane right where VP40 is located. At this point then, what we're going to find is this is going to start to protrude out from the cell. Okay, and if we go back to our original picture, you may recall that we saw this process of protrusion. These nucleocapsids are transported to the membrane along with NP and L, etc., and then we get some sort of budding and exit of the virus. But wait, the host cell actually has a way to prevent budding. One of the proteins that is induced by an interferon response is known as tethering. What is a tether? Anybody know what a tether is? Anybody play the most violent childhood game of all time, tetherball?
It's like a rope that keeps it attached.
Exactly, it keeps it attached, right? God, I love tetherball, except when I played against my dad, who was a bastard at that game. But my dad's incredibly competitive. All right, so tetherin is a protein that basically attaches or keeps a budding virus attached to that cell that it's on. In other words, the benefit of this is if tetherin is present, it prevents that virus from being fully released and having the ability to infect other cells. We'll see that HIV also affects tetherin levels. But here's what we find. Glycoprotein is actually absolutely required for exit, and it does so by somehow interacting with tetherin. So I'm showing you a couple of different Western blots here. This top one is looking at, the way that they've performed this experiment is they're looking at VP40 protein, and they're looking at the VP40 protein within lysed cells. So they've taken 293 cells, infected them with Ebola, let them grow for 24 to 48 hours. This is the amount of VP40 in the cells. This is the amount of VP40 in the media. And in the media, this indicates that these are released viral particles. And so they've labeled these as what's the VP40 concentration in the cells, and they've labeled this one just as virions. What happens as concentrations of tetherin increase?
VP40 stays inside the cell.
Yeah. There's less outside, maybe a greater abundance inside. In other words, there's more associated with the cells than not. This virus no longer has the ability to, what we would assume, bud or exit at this time when concentrations of tetherin are increased. Down here, what they take a look at are what are the proteins that are necessary for this. So again, they're looking for VP40, and they've basically created a number of different conditions in which they have a virus that's defective in one or more proteins, and then they're complementing these proteins back in. So for example, mock without tetherin, virus is able to escape with tetherin, and they're using concentrations that are equivalent to this highest amount. We get a block and exit. VPU is a protein from HIV. This is a positive control. When VPU is added in the presence of tetherin, what happens? Still gets some exit from the cell. Now they look at individual proteins from Ebola virus. GP, NP, VP24, and VP35. And basically, the way to do this one is which one looks like the positive control? Which ones look like the negative control? GP looks like the positive control. Therefore, GP must be required to help this virus overcome the inhibitory effects of tetherin. We don't exactly know how. It's probably a mechanism of cleavage, that GP may have some sort of proteolytic activity to cleave itself and the rest of the virus from tetherin. Jonah?
Do you know generally how tetherin works?
I don't, actually. This is one of the first times I've ever heard of the protein. But my guess is it probably binds rather non-specifically. But how it works, I have no clue. That sounds like a great topic to look at the end of the semester. How does tethering work? Okay, so that's replication and transcription in a nutshell, and everything through the end of the viral life cycle. So let's just, what I want to do real quick then is let's go back to the beginning. Let's take a look at this again. So we've talked about that this process of entry involves, we didn't necessarily say the protein, or we said the protein, but we didn't look at the evidence. TIM1 seems to be one of these proteins that is required for attachment, and GP attaches to it. We get some sort of receptor-mediated endocytosis or penocytosis to occur, and upon acidification of the endosome, what happens? How does the virus actually exit as the endosome is acidifying? Or how does it finish its entry and uncoding process? Well, this was a while ago, but Jonah?
The virus's membrane and the endosome membrane fuse.
That is true. Something has to happen in order for that event to occur, though. Drop in pH, and that activates what host proteins? There were two of them that we referred to. What was that?
GP1 and 2.
Those are the glycoproteins of Ebola that will get cleaved by this protein. Yeah, Cat B or cathepsin B and cathepsin A, or L, sorry. So those two are activated. They're in the endosome, but they're activated upon the drop in pH. They cleave GP. GP2 becomes revealed at this point and allows for this fusion event to occur. And at that point, the GP and the envelope remain as part of the endosome, and then the nucleoprotein L, VP24, 35, 30, I'm missing something maybe, but all of those proteins, 40, all exit here into the cytoplasm. This is kind of a misleading image, because this shows the negative single-stranded RNA, but it shows it unbound. And actually, this is going to be constantly bound by nucleoprotein and associated proteins. So here we've got nucleoprotein, VP35, L, VP30 driving transcription, VP35L and NP driving replication. Those negative single-stranded genomes plus those viral proteins begin to localize on the inner membrane. One that we didn't mention, VP24, is one of the minor matrix proteins, so it and VP40 help to recruit the scaffolding. And here's where we get that exit event. And tethering would be involved right about here. Okay. So that's the overview of the life cycle and how everything fits together. I'm going to pause and just see if there are any questions on what we've talked about today before we move ahead. Okay. One thing we haven't talked about yet. So we're going to talk about, kind of leading into the next bit of Ebola, is to understand how does it actually cause the disease that it causes. And we don't know all the answers. That's one of the hard parts is Ebola virus, the infections are rather rare. So we've only got about 40,000 cases worldwide to actually base our evidence off of, right? And it's rather sporadic, the cases. They pop up at periods of time that we can't necessarily predict. But we do know some things based on either some animal models or based on what we can observe in human cells, or just in cell culture. So we're going to talk about VP35, but I also want to talk about this SGP. And you may recall we said that this soluble GP protein here is produced as a result of some Slight misreading of the ribosome on the RNA. I'm not really worried about you understanding that mechanism so much, but I want to focus on what might be the role of the SGP. So, if you can, think about the immune. So you've got free-floating virus. Here's GP, my really crappy image. How do we block this, or how does our body block that from attaching to a new cell and infecting it?
Covering the spike proteins with antibodies.
Bingo. Covering the spike proteins with antibodies is one of the best ways, and we would call those antibodies neutralizing antibodies, is the technical term. Given that, what's the function of SGP, of the soluble GP?
Blocking those antibodies
In what way, Grace?
by binding to them.
Yeah. It's a decoy in some ways, right? So that soluble GP would bind to the antibodies and basically function kind of like a sponge. Soak up those antibodies so that there are fewer to bind to the Ebola virus genome. And if you remember, the ratio between SGP and GP, there's about 3 soluble GPs produced for every one full-length GP. So the virus spends a lot of energy of the cell producing this soluble GP. And again, the reason to do this is just to prevent a B cell response. Jonah, you got a question?
Could those SGPs trigger like an interferon response or something?
Yes, they could trigger some other response, absolutely. I mean, most likely they would be, for example, phagocytized by some macrophage and then allow for activation of CD4 positive T cells. That's likely. As long as they're free-floating like that, yeah, that's definitely possible. They may be inflammatory. I don't know the answer to that. And if they're inflammatory, that means that they're probably triggering cytokine release and histamine release. That might contribute to some of the effects that we observe as well. As a precursor to what we'll talk about next time we come back to this, kind of leave with this. VP35 is a multifunctional protein. And we actually find this not only, I mean, we find this in human cells to some extent, but viruses are really good at producing multifunctional proteins. Common core, but then there might be 2 or 3 different activities through their ability to bind to different protein interactors. And I've put up 2 examples of this. Why did I put up L2? I don't know, maybe I meant to put E6 there. But E6 and E7 from HPV are 2 of these examples. E7 not only binds to E2F, but guess what? Something we didn't talk about, it also binds to RB. It also binds to, or it's also going to, through its activation of E2F, going to activate the production of telomerase. And E7 has other binding partners as well. E6, well, E6 binds to what? What does E6 interact with?
E6AP.
E6AP, and that leads to degradation of P53. It also leads to degradation of other proteins. And E6 has other binding partners as well that we did not talk about. We can see this time and time again. Poliovirus has particular proteins that function not only as a protease, but also function as a polymerase at the exact same time, depending on how the protein is broken down from its polyprotein form to the individual proteins. VP35 is multifunctional. Consider it like a Swiss Army knife. Man, great analogy. All right? So you've got one knife, and then you've got the fishing hook, and you've got the saw in case you ever need to cut off a leg, and you've got something to file your nails if you really need to while you're out there too. That all Swiss Army knives come with. All right. So VP35 not only controls replication and transcription, but it's going to regulate the immune response. And it's going to do so through interacting with interferons. Not only is it going to repress the activation, but it's going to repress some of the downstream effects of interferon activation as well. This is a very brief overview of what interferons do. They are secreted by infected cells. Interferons, the major type, there are two types, 1 and 2. We're just going to focus on type 1, which sometimes you'll see the terms interferon alpha and interferon beta are the specific ones. But macrophages, other white blood cells are going to produce these when they recognize a substance as being foreign. So some sort of sign that infection has occurred. And remember, one of the cells that seems to be a host for Ebola virus is actually macrophages. So infection of a macrophage with Ebola virus will trigger this interferon response. It's going to activate some sort of signal transduction pathway, ultimately leading to the expression of interferons. And then those interferons are secreted and are involved in paracrine or autocrine signaling to help inform the cells around that area that, hey, there's an infection occurring. Begin to activate antiviral responses. That's basically the function of interferon. It's kind of like a beacon or a warning sign. Genes that are going to be induced or activated by interferon include some that we've actually already talked about. PKR. This is involved in blocking cap-dependent translation. RNA cell we haven't talked about, but this degrades double-stranded RNA. MHC1. MHC1 is a protein involved in adaptive immune response. And then finally, P53. An overexpression or activation of P53 can lead to cell death. And so what we find is that the cell surmounts this response to basically block a viral infection, and then if everything doesn't work out, it just kill itself as a way to prevent that infection from spreading. That's the main design here. We'll leave it at that, but I'm just showing you here. Here's where VP35 is going to be involved. We're going to see it blocking a couple of different proteins here, blocking specifically interferon beta, and then there's one more spot which doesn't show in here, but VP35's ability to block PKR directly as well. All right, so we'll pick up on this on, tentatively right now it's going to be Friday, but most likely it will be Monday next week. Plan to take a look at sequences sometime between now and Wednesday. I will begin to post those as they roll in, and I'll just put them under the Phage project as individual text files. All right, and I have your in-class exams if you want to grab them. You're not obligated to right now, but if you want to, you can.